Hello guys, back to English here. So I would like to welcome everybody, all the students, all the speakers again. Thank all the organizing committee and all the students. I would like to thank especially Dr. Julio, Cesar, Cetrulo, Lorenzi for accepting our invitation to speak here in our workshop to give a talk about his recent work that he's been developing in SARS-CoV-2 infection. So Dr. Julio Cesar Cetrulo Lorenzi got his degree in biology from the Federal University of Alfenas. And he later joined our graduate program in immunology, in basic and applied immunology, as a master's student. And I remember he joined the same time as me, right? So we joined it together for the master's degree. And later he moved on to the field of genetics, where he did his PhD, also both of them here at the Medical School of Ribeirão Preto in USP. And later he moved to the United States, where he worked at the Rockefeller University. He did his postdoc at the Rockefeller University under the mentorship of Dr. Michel Nussensweig, in which he worked mainly in HIV and also more recently in SARS-CoV-2 infection. And at present, he is a senior scientist at Moderna pharmaceutical company, right? Moderna that just got famous recently due to its RNA vaccine for SARS-CoV-2. So thank you again, Julio, and you can lead on from here and show us your results. Thank you very much.
Thank you, Diego. Thank you, Dr. Vania, Dr. Maria Carolina, and the whole committee for inviting me for this presentation. And it's really a pleasure to be here and to give the kickoff of this workshop. And as Diego was saying, I was back in Ribeirão Preto in 2006, also working and organizing a workshop in immunology. So it's really, really a pleasure to be here and to give this talk. So I'll share my screen and we can start this presentation. So again, thank you everybody for coming. And today I would like to talk about the evolution of immunity on SARS-CoV-2. And this is the final year of my postdoc, basically. And out of the beginning of this presentation, I'd like to say that this was a very large collaborative endeavor. Several labs at Rockefeller University and also collaborators elsewhere in Pamela Bjorkman's group in Caltech, Paul B. Nash also at Rockefeller, Rolf Barrick at UNC in North Carolina. And so this is a really great example of how collaboration can bring results in a very fast way when we have enough investments, both from the governments with different grants and also from different philanthropies that can really help push science in a very fast way when necessary. So I will present today results that compose several publications that were finished in 2020, 2021, before I moved to Moderna. So I don't need to give like a disclaimer here because I'm not talking about anything I did at Moderna. So just to start this presentation, this is the classical slide that I've been presenting many, many times in the past 2 years. This is the COVID dashboard from the Johns Hopkins University, which bring us numbers of infections, deaths, and nowadays the number of vaccinated individuals. It's really great to see that the number of vaccinated individuals is surpassed a lot already, the number of infected cases and also the number of deaths. And it's also good to see that Brazil is not in the top 10 list of cases or deaths. We still have a lot of new cases, both in the US and nowadays more and more in Europe, but we have a little bit better situation in Brazil. And that's probably due to the vaccinations that are massive in the past 6 to 9 months, right? So back in 2020, beginning of March, we started hearing about these viruses, and I'm sure this audience knows everything about SARS-CoV-2. but I'm just going to go over a very brief introduction about this virus. This is a coronavirus from the distributed in the coronavirus family as a beta coronaviruses. We know that this type of virus has been between humans for a long time. So in this slide, I show several examples of other coronaviruses that have different natural hosts, different intermediate hosts, and ended up being infecting humans. For instance, we have HCoV-229E, which is like a very old friend of humans being around for a long time and infect us. Also, we have HC43, and we have HQU1, which are what we call like seasonal coronaviruses that infect people all the time. But unfortunately, we have some other examples of coronavirus that became problems, like the two outbreaks we had before, the SARS-CoV-1, nowadays we call like that, and the MERS. So both of those viruses are also coronaviruses. One's based on intermediate host of a camelid and still infects camels back in Saudi Arabia, for instance, and there is a transmission from those camels to humans. If we go over the historical cases, we see that these two outbreaks back in 2002 and 2006 brought several hundreds of cases and some around 10 to 5,000 deaths, especially connected to the first SARS pandemic. That was an epidemic, sorry, not a pandemic, was more specific to the South Asia, especially Hong Kong and China. Which brings us to understand that this is not a new thing for humans, but unfortunately, this time we have a little bit more complicated coronavirus. So I bring here just a classical description of this virus, how this genomic organization and structure of this virus, the envelope virus, and we have here in the left side the spike in blue, the nucleocapsid protein inside the virus, and those are the most important proteins for the immune response. And fortunately, we have nowadays most of all the proteins already sequenced and with a very high detailed structure. And what that is important? Why we need a very detailed structure of antigens? That's really necessary for us to understand how B cells, for instance, can combat this infection, where antibodies can bind, and where we can have neutralizing activity that can prevent the infection of this virus to our cells. We know that this virus uses the ACE2 receptor to infect cells, and this receptor is expressed all over our body, especially in the lungs, but also in the heart, liver, colon, which explains some more severe symptoms like diarrhea and some liver and kidney problems that patients have once they achieve what we call the severe phase of this disease. And we see the secondary symptoms. Again, a classical review here of symptoms. We know most of the patients show fever, dry cough, fatigue, dyspnea, myalgia, and we have anosmia, which is when you lose the capacity of smell and taste. And that's in the beginning was a very important way of checking for infected patients before we had very good assays like PCR or ELISA-based assays for infection. In the bottom part of the slide, I'm showing the classical evolution of this disease. We have infection, which is mostly asymptomatic. 95% of the individuals that are getting infected have an asymptomatic profile of infection. Some patients will go to a mild, moderate response, which is correlated with a high viral load. And a small portion of those patients will become severe symptoms, and that is usually correlated with a hyperinflammatory response that can be resolved, but unfortunately in some cases can become unpractical and bring individuals to death. If we think about the immune response, this is a very simple cartoon about all the different possibilities we have for the immune response to really go against this infection. And one parenthesis here is that I think this is the virus that we really know most from all over the scientific history. There is hundreds of thousands of scientific publications about the infection and all the different possibilities of from the immune system to neurology to all possibilities to understand how this infection happens and what is really going on. And this is, I think, a very good thing because we really now have enough tools to understand this infection and maybe come up with better treatments and better vaccines. Today, we're going to be focusing on the B cell response, especially for antibody secreting B cells, how they come up after infection, and how affinity maturation happens in this type of infection. So the main aim of this study, which was a direct reflection of this new pandemics at that stage back in March 2020, was to repeat something Michel's lab has been done for different viruses like HIV, hepatitis B, and more recently TEBV, which is trying to find patients that have a very strong and specific response against infection and can make antibodies that are strong neutralizers that can really prevent infection, neutralizing the virus to infect the target cell. So we have data, and I participated in some of those publications before, showing antibodies that can prevent infection for HIV in vitro, for instance. In those antibodies right now are also licensed for pharmaceutical companies that will use that as maybe as a treatment in the long term. So the very bird's eye understanding of our protocol here is a regular screening. We have a hospital at Rockefeller University. We have an IRB approved protocol, and we start recruiting individuals that had been infected and were already healthy, let's say, after a while, after they didn't have any viral load activity. So we recruited those patients and start screening by ELISA and by neutralization assays, and selecting those patients for large blood draw experiments where we were able to obtain millions and billions of PBMCs actually that we further analyzed and selected B cells for single cell antibody cloning. And I will go over this in more detail later. But just to make clear here, this is a protocol that could be repeated in different places, depending mostly on collaboration and investment. If you have, for instance, at the medical school in Ribeirão Preto, we have enough labs, enough brains to do it, I think. We just maybe need the money to do something very similar whenever we have a different outbreak. So that's one message I would like to make also. It's not about being outside and with different scientists. It's more about just planning and being able to perform this type of experiments in a fast way. So just going a little bit more fast in this, we did this recruitment and we ended up recruiting 149 individuals in this first stage of the study. The first reagent that we obtained, and I think is a very important reagent, was the recombinant RBD. And that's the specific region in the spike protein of this virus that is necessary for interaction with the ACE2. So two details here. First is that the sharing of the genome sequence of this virus back in January 2020 by the Chinese government, that was a very important move because that made possible many labs all over the planet to make these recombinant proteins and start testing patients. So once we had this recombinant RBD, we started doing ELISAs, right? Classical ELISA, just to titer different antibodies from these individuals. And here I start showing a little bit more of data, and we can see these panels from the left to the right. First, ELISA titers against the RBD, and in the bottom part, ELISA titers against the full spike. And in blue and red, I'm just exemplifying here two strong responders, patient 21 and 47, that will be important later in the presentation. You can see that most of the patients have a very strong IgG and IgM response, and the IgM was lower as expected since these patients already were controlling this infection after the viral load was reduced to 0. If we then sub-select this patient and check if they were hospitalized at some point during the infection, we see that those patients had also a higher titer against the full RBD in the IgG compartment. And we see also that males have higher titers than females in this case, and that was a result that was further repeated in several different labs. In order then to test those serum samples for neutralization capacity, what I did with collaboration with Frau Merckx, which is now a PI back in Heidelberg, is to create a system using HIV-based pseudoviruses, where those viruses will express the spike protein from the SARS-CoV-2 virus. So we have a recombinant virus that can infect cells that are then express the receptor. So for that, we created 293 T cells that highly express the ACE2 receptor. So if you combine this pseudovirus that carries a nanoluciferase reporter gene, you can then detect infection by the activity of that reporter gene by luciferase. If you then combine these two reagents with the sera from the patients, then you can provide a very detailed and let's say a precise measurement of the NT50s, which are the number of sera necessary to prevent 50% of the infection. And this will be like the workhorse of those experiments. So first we selected all the patients from this step in the trial and start testing all the sera against our pseudovirus-based neutralization. And we saw already a very interesting result. We saw that most of the patients, the bottom 79% of all these patients, had an IC50 that was below 1 in 1,000. What that means that most of the infected patients made a very low activity pooled IgG response, but in some cases, in 2% of those cases, we saw responses that were higher than 1 in 5,000, which means we need a very little amount of sera to neutralize this infection in vitro. When we then correlate the levels of neutralization with the levels of IgG, we saw a very strong correlation as expected. And again, patients that were hospitalized had a higher capacity of neutralizing infection in vitro compared to the other patients. We then went for our antibody campaign, which means we start, instead of testing sera, now we start testing cells from these patients. So we were able to obtain a large amount of blood from these patients, like usually around 200 mLs of blood. And from that blood, we obtained PBMCs and went for our classical single cell sorting strategy. So I think that's important for this audience. We see in the left side, the very last panel of our cytometry, where we do a single cell cloning of these B cells. And here we use two different fluorophores for the RBD. So that's why we have an Alexa fluor 647 and the PE. And that's a kind of is important detail. It's important to have a true color binding for selecting specific B cells. Because as you can see in the lower right side of this graph, there is cells that binding to only the PE fluorophore RBD, which probably is not a very good cell. So we only selected the ones that were double positive in this case. Those cells were single cell sorted, and then we did our classical multiplex PCR, where we are able to have PCR results for all heavy and light chains using a mixture of 51 different primers. Using those primers, we're able to clone those heavy and light chains in different plasmids. We use those plasmids to transfect 296 E cells. And from those transfections, we're able to obtain purified monoclonals now from the supernatant. So that's our classical pipeline to obtain monoclonals from B cells that were specific for RBD. And here I just brought a little bit more detail because I know there's some cytometry fans in this audience. And it's mostly a very simple gating process where you get lymphocytes, single cells, you dump channel for CD3, CD8, CD16, CD14, and a zombie for live and dead cells. You then select CD20 positive cells, B cells, and from there we also stain for irrelevant antigen like OVA in this case. And from the negative cells, we then run our double gating P-Alexa647 for single cell selection. Once we did this, we obtained the sequences of those heavy and light chains. And in these pie charts, what I'm showing is in the middle is the number of single cells from each patient. So for instance, patient 47 had 79 B cells sequenced. And the colorful slices in these pie charts represent sequences that are shared, the heavy and light chain genes on the VDJ. So every time you see a color that repeats, it means that in different patients, we have similar VDJ genes recombined in the same population. And it's also interesting to see that 32% of the response in this case for RBD was clonal, meaning that we have B cells that expanded and could provide an affinity maturation started for this response. If you go a little bit more in detail, which I think is cool, if you go even to the most variable regions, the CeTI-RP, we see that for different patients, for instance, patients 21, 57, and 107, we have the exact same V, D, and J, and even the CeTI-RP is very similar with only single nucleotide mutations in that region, which shows that different patients responded similarly for the same infection, which is pretty cool and not really expected. If we then compare our data from our paper with different papers here in this publication from Dennis Burton team, also in 2020, they show that in different manuscripts that screen different populations, the type of recombined B cells were also very similar. And the specific V353 was the most predominant B cell over hundreds of individuals in this analysis, where all of these different studies perform also single cell cloning. And the team of Dennis Burton and Ida Wilson provided a hypothesis for this, which was based on the structure. Whenever you bind that antibody that it has that specific heavy light chain combination with RBD, you see that if you have the specific CeTI-RP, that is necessary for a protrusion that enters in the ACE2 binding pocket. So in the left side, in the lower left side panel, we see an example of a positive correlation where that specific VDJ can really go in that pocket and bind. And the right side, we see a different VDJ that cannot go to that region. And the conclusion of this article from Burton's team was that that specific recombined heavy and light chain gives the opportunity for that antibody to really be selected positively and go to the next rounds of affinity maturation in Geminal Center. So once we finish that first analysis on the sera and sequence the cells and obtain the monoclonals, we start then testing individual monoclonals for, again, ELISA titer, NT50 titers, and even real infection using SARS-CoV-2 viruses instead of pseudovirus. And I give here three examples of monoclonals, which are monoclonals 121, 135, and 144, that have a very strong activity against this infection in vitro, against both pseudovirus and a regular real virus infection, with IC50s below 10 nanograms per ml, which is a very good titer for monoclonals. And we did several collaborations to profile those three candidates. So Pamela's Bjorkman lab did the structure of binding of these different antibodies. And here again is a very interesting point of observations. Each of those three antibodies bind differently in the RBD, in different regions of RBD. And that's very important if you think about doing a combinatory treatment that uses those antibodies that will prevent escapes from antibody selection, right? If you treat those patients, if you have a high viral load, maybe you're going to select RBDs that can escape those antibodies. But if you have a combination of antibodies binding to different regions of the same protein, that chance decreases drastically. Another collaboration we did with Paul Binash lab that I really kind of pipetted these experiments. We did an in vitro evolution experiment where you superinfect these cell lines with several passages of pseudovirus, and then start treating those cultures with high levels of antibody until you start getting escapes. And we found escapes pretty fast after 5 passages. And the escapes we found were very similar to the VOCs or the variants of concern that we see now in the real population. Those are the expected escapes, let's say, in regions, for instance, in the 484 E2K variable, which is something is in the P1, for instance, here back in Brazil, but also in the variable that came from the UK. And later on, in the end of the year, we did like an in vitro model using the golden hamster to show a therapeutic effect of the combination of using those antibodies that we selected from our individuals back in Rockefeller, and show that the combination of those antibodies in vivo provided the neutralization and protection against infection in animals, providing evidence that we could maybe use those antibodies for treatment in humans. So we have a very long summary here, but I think the most important part is that potent monoclonals can be isolated from all patients. So besides most of the patients having a low to mid response in the sera, if you go for single cells, we always can find very good examples of monoclonals. And that's why I think most of the patients that are infected are asymptomatic, because they can make really good response, even in a small portion of the B cells. So then we did a follow-up study 6 months after the initial infection. And I'm just gonna go over this also pretty fast, because we saw that most of the patients in a serum activity had a reduced activity both by titer and anti-50. So the overall activity of those sera was lower. But if you go back and start sequence the cells from the same patient 6 months after the infection, we see first in the top right side of the slide on the B that the number of RBD-specific B cells is actually maintained. It's even a little bit higher, let's say. Whenever you sequence single cells, we see a higher level of somatic hypermutation in all of those cells. So if you go down B, you see that if you compare 1.3 months and 6.2 months, the number of mutations accumulated in the heavy light chain goes from 10 to almost 15, which is a very important detail showing that this response is evolving. And we also saw that these cells were expanding specifically in clones that were highly potent against the RBD. If you then check for specific clones, and overall we see that clones that evolved in the 6 months had also a higher titer, meaning that they were selected for affinity maturation. And if you then go and test those antibodies against the different variants of concern, so then just bringing here a recent paper showed all the different number of lineages from SARS-CoV-2 from 2020 all the way to May 2021. So in the panel C here, we see that we went from the Wuhan wild type, to more than 1,200 different variants nowadays. So our lab went through and did 41 new spike constructs with all these different variants that we obtained back in September of 2020. And that's a lot of work. So I just brought here one photo of showing all the different viruses that we grow for this experiment. But that was a very cool experiment, because then we tested all the different monoclonals we had. And in this figure, if we start from the bottom to the top, in black is the representative of a specific monoclonal we obtained in the first time point, 1.3 months after infection. And in red is the name of that same clone that evolved 6 months after infection. So we tested 48 pairs of antibodies against, in this case, we're showing 10 different variants. And we see that in most of cases, the antibodies become better, and the affinity maturation really brought these antibodies to be able to detect and neutralize variants of the SARS-CoV-2. And here just one example of 3 different pairs, and in all cases we see an enhancement of activity from inspecific, for instance, on antibody 144 to super specific on antibody C051, which are a pair of antibodies that can even neutralize the 484 variant. So just one example of one pair of antibodies that now are able to neutralize some important variants of SARS-CoV-2. And all those examples are important to show that affinity maturation is really able to push a better antibody if you have stimuli and if you have germinal centers that are working against that infection. So the summary here for the 6 months after infection, that we see persistent of shared clones, we see increased number of mutations and no decay in the number of memory B cells. And then after I left Michel's lab, they did a 12-month follow-up, which is just 2 slides, but I think it's also cool to show that if you go from the left to the right, again, we see 1.3, 6.2 months, and 12 months. And also, some of the patients that are originally infected also got vaccinated. And those are the blue dots in all of those dot plots. So if you focus on the right top corner in the D part of the figure, we show here the NT50 of those samples in the sera, in this case, from 1.3, 6.2, and 12 months after the original infection in patients that were vaccinated. And we can really see here the effect of the vaccination in those patients where there's a really great bump in activity of NT50 from 83 geometric mean to more than 3,000 geometric mean, which is compared to the C panel showing that the change, if you did not get a vaccine, just persists between 80 and 75 NT50, what shows that you can keep your activity over time, but if you get a vaccine, you really boost that immune response. And that's what we expect, right? Because you're just pushing a ton of more antigen for those cells to really activate and go to germinal center and push for affinity maturation. So I'm not going to go over all of these details because I want to have some time for discussion. And the summary for this third part is that after 6 to 12 months of infection, individuals that get vaccinated have a profound boost immune response that expands specific RBD memories to the B cell pool, and that provided a further stimuli for maturation. I didn't show here, but if we went through all those specific clones that evolve over time, we saw that that boost also expanded further those clones. And that for sure pushed this affinity maturation that I was mentioning. So I think this is a very clear and interesting example of how our immune response can be super strong against a new pathogen, and how could vaccines really help on boosting originally our own immunity against those pathogens. So I would like to thank the whole lab for helping with this. This is like a 60 people lab. There's a lot of collaborators, and I'm open for questions. Thank you.
So thank you very much, Julio. It's extremely interesting to see all this amount of data that you guys have generated there on COVID, like in 2 years that we have the disease going on. It's amazing, you know. It's really amazing what has been done in this area.
Thank you.
So I'm going to switch to Portuguese now because I'm going to read some questions here.
Sure.
Então, o Vinícius tem uma questão aqui. O laboratório Fleury divulgou na semana passada a comprovação da eficácia da vacinação no combate ao Covid-19. Divulgaram vários tipos de anticorpos entre eles os que chamam de anticorpos básicos e neutralizantes. Os anticorpos básicos seriam aqueles que a pessoa tem quando adquirem a doença. Poderia explicar melhor sobre esses anticorpos?
Eu não vi falar sobre esses resultados, Vinícius, eu prefiro não comentar. Talvez o básico seria da primeira infecção, comparado com o que é posterior à vacinação e depois da maturação da afinidade, mas eu não fiquei sabendo sobre isso.
Tá certo. Eu tenho umas perguntas em que eu fiquei curioso aqui, Júlio, também. Vocês fizeram essa seleção de anticorpos neutralizantes, né? Eu tinha visto já alguns trabalhos do Nussenzweig fazendo isso para HIV e tal, anticorpos altamente neutralizantes. Vocês também fizeram essas bibliotecas de clones aí que vão neutralizar os coronavírus. Eu achei interessante quando vocês forçaram a infecção in vitro, as mutações que aparecerem serem muito similares àquelas que são detectadas nas variantes de preocupação, aí no caso. E eu queria saber se você tem mais informação sobre, eu acho que isso já deve ter sido feito, estudos em cima de quais as regiões da proteína spike, por exemplo, que são mais conservadas e não sofrem tantas mutações, e teriam clones específicos para essas regiões que teriam atividade neutralizante tão potente quanto os anticorpos que vocês observaram, por exemplo.
São duas partes dessa resposta. Se você olhar across diversos artigos que fizeram single cell cloning, tipo, por exemplo, o professor Mike Diamond, do próprio Ian Wilson, o pessoal da Holanda também fez diversos artigos. No começo, todo mundo achou anticorpos naquele pocket da região, que vai de 420 até 490, mais ou menos, que são os mais induzidos pela resposta original, let's say, e também eles são os que a gente selecionou via os nossos experimentos, né, que é só RBD. Quando a gente começou a fazer spike, no lab, por exemplo, a gente viu que era muito mais difícil obter spike estável para fazer sorting. E ela era muito mais sticky. O que é isso? Quando você começa a sequenciar, você vê que você está pegando anticorpos que não tem nada a ver. Então, a partir de um tempo, no começo desse ano, na verdade, tiveram alguns outros estudos que estabilizaram o spike melhor e mais anticorpos começaram a aparecer. E tem anticorpos hoje com fentomol de afinidade. Alguns deles em região de base do spike, por exemplo. Na região de 650, por exemplo, tem diversos anticorpos naquela região. E enquanto a conservação, tem um artigo da professora...
Eu fico com essa ideia, essa dúvida, na verdade, quais mutações não seriam deletérias, né? Para o vírus perder a capacidade até de infectar. Se ele tem uma região conservada nessas proteínas, que poderia ter algum, principalmente se for trabalhar com monoclonais. Né?
Então, por exemplo, quando a gente fez a co-culture, eu estava falando, com o evolution in vivo, quando você tem as combinações das diferentes regiões, você não acha variante. Você não consegue. Você tem que fazer subsequential. Você começa com um anticorpo, ele evolui e cresce. Aí você põe um segundo, terceiro, aí você consegue encontrar as variantes. Mas se você tem... O mesmo acontece em vivo, né? Se você tem uma resposta policlonal, é muito difícil você fugir. E eu achei interessante que a gente achou essas mutações lá, como a 484, logo de cara, porque parece que ela é uma região que não importa para o vírus, em termos de tipo flip-flop. Você pode mudar o aminoácido, que ele continua infectando igual, entendeu? A Delta, por exemplo, eles têm uma variação na base do spike que é muito importante e causa uma maior capacidade de infecção, em termos de célula mesmo. Quantas células o vírus... Tipo, quanto você precisa de vírus para infectar uma célula. Mas que não mudou em termos de severidade, na verdade. Né? A Delta não é uma coisa mais severa, uma infecção mais severa, ela é uma infecção mais ativa, vamos dizer, com um viral load mais alto. Então aí tem alguns anticorpos que o pessoal já está vendo nessa região e os anticorpos que foram induzidos pela vacina são muito semelhantes. Isso eu também achei muito legal quando a gente viu esse resultado. Que a vacina realmente mimetizou a infecção, mesmo em pacientes naive, né, não infectados.
Legal, muito obrigado. A professora Vanessa levantou a mão aqui para fazer uma pergunta. Você quer perguntar no microfone, Ivan?
Oi, Júlio, parabéns, muito lindo o seu trabalho, viu? Obrigado. A minha pergunta é sobre, você sabe se algum desses clones pode apresentar, ou se você checou, cross-reatividade a outros vírus, principalmente vírus de RNA?
Não coronavírus, você fala?
Poderia ser o SARS-CoV,
Não, a gente fez todo um painel, por isso que eu coloquei 41 variantes. A gente fez todo o painel de bat, de pangolim, de tudo que tinha basicamente na literatura de coronavírus. A gente só achou um anticorpo com cross-reactivity boa, mas ainda é raro. A professora Bjorkman, ela fez uma estratégia vacinal bem interessante. Ela usou uma ferro nanoparticle, que você coloca centenas de RBDs ao mesmo tempo nessa, como se fosse um droplet lipid, um LNP, né? Só que é de ferro. E ela fez uma maneira que você coloca variantes de todos os coronavírus endêmicos ao mesmo tempo. E eles conseguiram gerar uma resposta muito potente, que é cross-reativa. Porém, ela não é o bastante para pegar as novas variantes de concern. Então, existe uma separação aí meio grande do SARS-CoV-2 para os outros coronavírus.
E sobre outros vírus respiratórios, existe alguma possibilidade? Porque acho que agora vão aparecer algumas doenças imunomediadas por excesso de anticorpos, talvez até.
Eu não imagino que isso seja possível, porque a RBD é muito específica ali desses coronavírus. Em termos, se fala de ter uma cross-reactivity para outros tipos de spike, por exemplo, né? Não que eu saiba, por exemplo.
Tá certo, obrigada, viu?
Que isso.
Júlio, tem mais duas perguntas aqui. O Vinícius mandou uma outra pergunta, se há perspectivas de aplicações dessa plataforma na produção de anticorpos ou uma parecida para outras infecções virais, que não de SARS-CoV-2?
Sim, tem diversos laboratórios e empresas fazendo isso. Por exemplo, a Abcelera é uma empresa que é especializada em fazer monoclonal de qualquer coisa que você imaginar. E eles têm uma pipeline só para infectious diseases.
E tem uma pergunta aqui da professora Demilson também, que como as quase espécies virais podem impactar no tratamento com esses anticorpos anti-RBD e vacinas?
Isso é um exemplo muito legal, legal entre aspas, né? Dos anticorpos da Regeneron, que são os anticorpos que o Donald Trump tomou, né? Quando ele ficou doente. O par de anticorpos que eles escolheram, ele caiu com a 484, por exemplo. Ele não funciona, ele perde completamente a atividade. Então, aqueles primeiros coquetéis, o da outra empresa, não lembro agora o nome dela. Nossa, estou esquecendo as coisas. Mas tem duas empresas da Lilly. A Lilly também já foi aprovado. Aquele par de anticorpos ainda é ativo, mesmo nas variantes de concern. E tem novos projetos de monoclonais. Porém, as vacinas estão aí e elas são muito mais eficazes e mais baratas, né? Então, eu imagino que o mercado para monoclonal vai ser muito seletivo para imunodeficientes, para idosos e talvez para trabalhadores da área de saúde que precisem entrar em regiões onde tem uma carga viral muito alta, preventivo mesmo.
Tá certo. Tem mais duas perguntas aqui, a gente ainda tem tempo para mais algumas perguntas. Finalmente os alunos começaram a perguntar, né?
Ah, tá. Você não quer depois fazer a transição, Diego? Eu posso até as duas, se você puder.
Você diz para...
Da transição para a indústria, que você me perguntou para a gente falar um pouco sobre isso.
Ah, tá. Isso é uma coisa que seria interessante de você falar alguma coisinha sobre isso, né?
Mas termina essas duas perguntas e a gente faz isso.
Vou fazer isso aqui rapidinho aqui, então. Ó. O fato de as vacinas aprovadas para uso até o momento serem imunizantes não esterilizantes é algo positivo, uma vez que se tem uma maior dificuldade em selecionar variantes infecciosas? Essa é a pergunta do Samuel.
Essa questão é interessante, porque vacina esterilizante é uma coisa meio rara, né? A gente não tem muitos exemplos. E, do meu ponto de vista, as vacinas são muito eficazes e as variantes de concern, elas podem aparecer, lógico, a gente já tem um exemplo muito clássico disso, que é o HIV, né? Se você parar de tomar os remédios, você acaba selecionando novas variantes no seu próprio corpo. Mas, nos casos do SARS, se a gente chegar numa cobertura vacinal aí de 95%, eu imagino que a gente vai ter um controle muito bom, né, da doença. A gente ainda não está lá, mas a gente está chegando lá, né? Então, Eu penso mais nesse lado.
Tá certo. Tem a pergunta do Anderson aqui também, se há alguma evidência sobre a presença de um enhancement dependente de anticorpo frente a infecções posteriores com outras cepas de SARS-CoV, talvez como acontece com infecção com dengue, por exemplo.
Isso teve milhares de artigos, sério, era um problema muito conversado no começo, mas ninguém mostrou antibody dependence enhancement em vivo, especialmente o pessoal que estava testando, no princípio, o tratamento com plasma convalescente, né, que até o professor Gil Santos aqui da medicina fez bastante testes, e eles têm alguns resultados bons, mas em general, os resultados não são tão fortes, né, tem uns resultados de mais de 1.500 testes e não foi mostrada uma diferença estatística contra os melhores tratamentos, infelizmente. E é esperado, se a gente olhar os resultados, porque em geral a resposta é baixa. Você tem células boas, mas o título daqueles anticorpos não deve ser muito alto. Então, o ideal seria selecionar os melhores respondedores e daí sim usar aquele soro como um tratamento, mas eles são raros. Né?
Tá certo então, Júlio. Uma coisa que eu tinha pedido para o Júlio talvez dar uma ideia para os alunos aqui que é interessante, é justamente isso. Toda essa pesquisa que ele desenvolveu aí em SARS-CoV-2 foi dentro da área acadêmica, né? Dentro do laboratório do professor Michel no Senzoi, e agora ele transitou para a Moderna, que é uma empresa farmacêutica. Então, a gente está meio em cima da hora aqui, Júlio, mas se você puder só dar uns insights, assim, o que muda assim, quando a gente vai para a área privada, fazer pesquisa dentro da empresa?
Eu tive muita sorte de entrar num lugar onde tem uma pesquisa básica muito forte e hoje em dia eu voltei a fazer o que eu fazia no meu mestrado, que é trabalhar com vacina, né? E eu sinto a necessidade muito grande de gente, a gente faz, como fala, processo seletivo toda hora e é muito raro encontrar boas pessoas que querem vir para a indústria. E a indústria basicamente traz um foco, acho que essa é a maior diferença. Você tem uma pergunta que você tem que responder e você tem que chegar a uma resposta em um certo tempo e um certo budget. E se isso não for para frente, você vai acabar indo para outra pergunta. Às vezes você não tem o tempo de destrinchar todos os porquês daquela pergunta que você fez não ter funcionado. Mas você vai tentar responder outra pergunta. Então você tem toda a capacidade de gerar novas respostas científicas que vão acabar virando um produto. Né? Então acho que essa é a ideia. Talvez, eu não sei como está hoje no Brasil, mas antigamente existia certo preconceito de mudança tal, que não é tão legal, mas eu vejo pelo contrário, que a indústria ainda é um caminho muito interessante para a pesquisa e espero que no Brasil sejam abertas novas oportunidades também para esse lado da pesquisa.
Eu acho que um grande problema nosso aqui é a falta de oportunidade nessa área. Né? Tem poucas empresas que desenvolvem pesquisa e desenvolvimento aqui ainda, que pode absorver, convenhamos que é uma grande quantidade de recursos humanos que a gente gera aqui no Brasil, mas que ainda é majoritariamente absorvido pela universidade, a indústria não absorve.
Eu acho que os dois lados disso são investimento, tanto público quanto privado. Se você tiver investimento e você começar a fazer spin-off dentro da universidade, é assim que começam startups. E a partir daí você começa com uma ideia e ela pode ir para frente ou não. Então você começa de novo. E acho que precisa de investimento, não tem outro jeito.
Então tá certo. Vou passar para o inglês novamente. So I'm going to switch to English again. And now I'd like to thank Julio a lot again for accepting our invitation to speak here at our workshop. So it has been a pleasure for me to see you again and to talk to you again, right? So thank you very much, Julio. Your results are amazing, and all the things you showed to us here, all the information you gave to us here, all of it is amazing. Thank you very much.
Thank you, guys, and have a good workshop. Really enjoyed this time. I really remember my experience, so those workshops were really, really great. Unfortunately, it's not like alive. We don't see each other, but still try to get as maximum as possible from the speakers and also the poster sessions, the oral sessions. I know they are very interesting. So please enjoy. And I'm open to connections if anybody wants. I'm on LinkedIn. And if you have an idea, want to come to US, we're hiring. There's 650 positions open in Moderna right now. So talk to you guys soon.
Right, thank you, Julio. Nice to see you.
Thank you, guys. I have to go also. Bye.
Okay, thank you, Julio. So just remembering everybody, now we're going to start the next session at 3:55, right? So that's going to be the oral presentations. So Daniel, that's it, guys. Thank you very much for all of you that followed us. Bye.